Hi, welcome to my video on the Calvin cycle. The Calvin cycle, also known as the dark reactions of photosynthesis, is the part of photosynthesis that most of you seem to be struggling with. So hopefully this video will help you better understand what you need to know about the Calvin cycle. There's 3 stages to the cycle, and these 3 stages you need to understand. First stage is carbon fixation, the second stage is reduction, and the final stage is regeneration. In this video, we're going to look at each stage in a little more detail. Hopefully that'll help you understand what's going on and what you need to know. Okay, so you start with carbon fixation. Carbon fixation basically means you're taking some carbon from carbon dioxide, and that carbon dioxide is in the air and in the water inside of the plants, dissolved in the air, dissolved in the water. It's just there. Okay, you're taking that carbon and you're taking the carbon atom from carbon dioxide and you're adding it to another molecule. You add it to a molecule called ribulose bisphosphate, or RUBP. Alright? RUBP is a 5-carbon molecule. Once you add the carbon from carbon dioxide, it becomes a 6-carbon molecule. Okay? So essentially, you're grabbing carbon out of the air so the plant can use that carbon later to build glucose. Alright? An enzyme called ribisco makes this happen. Once that 6-carbon molecule is formed in a chemical reaction catalyzed by ribisco, almost immediately it breaks apart into 2 3-carbon molecules. And the abbreviated name for these molecules is PGA. So you got 2 PGAs. One of the carbons in those PGAs came from carbon dioxide. The second stage is reduction. Now, in chemistry, reduction describes a type of reaction in which a molecule gains electrons. Okay? And usually in the process of gaining those electrons, chemical bonds are formed, or chemical bonds are changed in some way. Now, what happens in reduction in the Calvin cycle in photosynthesis is essentially what you're doing is adding energy to the PGAs you create by adding electrons from ATP and NADPH. Okay? So the chemistry that's happening here is chemistry that takes you from a low energy molecule to a higher energy molecule. Okay, we're going uphill. What you get at the end of the reduction phase is a molecule called G3P. It still has 3 carbons, but now the bonds in between those carbons, the bonds in those molecules have stored in those bonds the energy from ATP and NADPH. And if you remember, the ATP and the NADPH were made in the light reactions. So essentially you're taking the energy that the plant has captured from light and you're storing it in chemical bonds in these G3P molecules. Okay? Now, if you look at the diagram down here in the left corner, it shows 6 carbon dioxides entering the cycle, forming 12 PGAs, which are then reduced to form 12 G3Ps. The reason the diagram shows it this way is because in order to get enough carbon into the cycle to build a glucose, 6 carbon dioxide molecules have to enter the cycle. Now, that takes us to the next stage, which is regeneration. What's being regenerated is the molecule you start with, the RUBP. You need that molecule back in order to start the cycle again. So once you have 12 G3Ps, and here's 6 of them, you've got 6 new carbons in the molecules in the plant. All right. If you look at the molecules shown on the slide, the blue ones, the blue C's, indicate carbon from carbon dioxide, and here's the other 6. So once you've got 12 of them, you've got enough carbon to build a glucose. So what happens is 2 of those G3Ps exit the cycle, As you can see down at the bottom of the diagram, 2 of those G3Ps go on to form glucose or other organic molecules. The regeneration part of the Calvin cycle refers to taking the rest of that carbon and rebuilding RUBP. Okay? So at the start here, you're essentially starting, if you have 6 RUBPs, you're starting with 30 carbons. You add 6 more carbons from carbon dioxide, that gives you 36 carbons. You energize those molecules, again, you have still 36 carbons, but now you can take 6 of those carbons out and you can build something with them. You can build glucose, which is the whole point of the Calvin cycle. Using the energy you got from the light reactions to make a sugar molecule. And the regeneration stage refers to taking the rest of that carbon and rebuilding those RUBPs so the whole thing can happen again. And again, this also takes energy. You're adding energy, again, energy that came from the Calvin cycle, or sorry, from the light reactions to do that. So, 12 G3Ps, 36 carbon atoms, 30 you got from the initial RUBP, 6 from CO2. 10 of those are used to make more RUBP, and 2 of those G3Ps go on to make glucose. So overall, making one glucose molecule requires 6 carbon dioxide molecules, 12 ATPs, and 12 NADPHs. In addition, you need another 12 ATPs in order to rebuild the RUBP 5 carbon molecules you start the cycle with. Okay, so to recap, step 1, fixation, grabbing carbon from the air, join it to existing carbon-based molecules. Step 2, reduction. You're energizing the molecules you've made. You're adding energy from the light reactions to your PGAs to create G3Ps. Step 3, regeneration. Some of the carbon exits the cycle, but the rest of it has to go back and build those RUBPs again. Okay, that's the Calvin cycle. Hopefully that helps.